This is part 3 of chapter 3, Bioenergetics, of our anatomy and physiology text. In part 2 we were talking about some chemical reactions, and we were getting into the idea that all chemical reactions in your cells, which constitute what we call metabolism, are catalyzed by protein enzymes, and that the activity of those enzymes depends critically on the shape of the proteins, which are subject to the right range of pH and the right temperature range. In this section, we're going to start talking about the actual formation of ATP. We did mention that the reactions in cells with respect to energy are coupled, and that we have exergonic reactions releasing energy as we oxidize fuel molecules, coupled driving endergonic reactions in which we make something more complex out of something similar, or at least with a higher energy content. And ATP was the simplest example that we gave at the end of the last section. So, how do we make ATP in the cell specifically? Where does it come from? Where does ATP for contraction come from? The simplest source of ATP is that dissolved in the cytoplasm, and when you begin a muscle contraction, then that's where the ATPs come from for the cross-bridge cycles, as we'll see. As soon as ATP starts to become exhausted within the cell or utilized, phosphocreatine is utilized to replace ATP. Phosphocreatine is another high-energy phosphate-containing energy storage molecule, which can then be used to recapitulate ATP for further muscle contraction. Then, if we need more ATP, then we get into oxidation of glucose, a 10-step process called glycolysis, which takes place in the cytoplasm. Typically, you have glucose stored in the form of glycogen in the muscle cells. You start hydrolyzing that glycogen, releasing glucose, and then glucose can be oxidized, producing a small number of ATP as well as pyruvate acid. And then ultimately, if we need more ATP than, say, a minute or two of exercise by glycolysis can produce, we will undergo oxidative processes in the mitochondria to produce a large number of ATP per glucose. So let's take a look at the anaerobic pathways. When we say anaerobic pathways, we're talking about those phosphocreatine breakdown to recapitulate ATP and glycolysis that we mentioned. Glycolysis done in isolation is an anaerobic process. It doesn't take any oxygen to run through those 10 chemical reactions by which glucose is converted to pyruvate acid, giving us 2 net ATPs, as well as some high-energy electrons, as we'll see. NADH is an electron carrier. That's something else that we discovered in the last part of this chapter. Oxidation of fuel molecules produces the release of high-energy electrons, and they must be captured, they must be transferred in an oxidation-reduction reaction to a carrier, to another molecule. In this case, there's a carrier called NAD, which will capture those electrons and hydrogens from the glucose as we oxidize it. Aerobic pathways occur in the mitochondria and involve oxidative phosphorylation, Kreb cycle and oxidative phosphorylation. So we'll define those things and look in a little more closely at those in this section. The main purpose of Kreb cycle is to gain electrons, again, high-energy electrons, as we further oxidize the pyruvate acid from glycolysis, and so we wind up with NADH and FADH, which are, again, electron carriers, and we can capture those high-energy electrons and deliver them to the electron transport system to make a lot of ATPs. So looking even a little more closely in at the anaerobic processes. So ATP is kept in the cytoplasm at a certain homeostatically determined level in resting cells. And when we start to exercise, begin to deplete those ATPs, and immediately phosphocreatine, which is also maintained at a set base level in the cell, is used to reproduce ATP. So here you see When we've used ATP, then we have ADP. ADP plus phosphocreatine produces ATP once again. And then we have the creatine molecule, which must be re-energized, rephosphorylated. That can last for about 15 seconds of exercise. If we still need to exercise for a longer period of time or choose to do so, we'll need some additional ATP, and we'll take glucose molecules and run them through, again, a series of 10 chemical reactions we call glycolysis, and wind up with 2 pyruvic acids and 2 ATPs per glucose. In order to do glycolysis, we're going to have to invest 2 ATPs initially, and then we will obtain 4 ATPs per glucose ultimately, and the net number of ATPs, which is usually what we mention, is 2. So for every glucose molecule that runs through glycolysis, we get 2 net ATPs. We also get 2 NADHs, though, high-energy electrons on this carrier, and pyruvic acid or pyruvate, 2 pyruvates. So if we operate in isolation with glycolysis, here I've written out the reaction in more detail. This is a more balanced chemical reaction describing glycolysis. Glucose plus 2 NAD, we need those carriers, plus 2 ADP yields 2 pyruvic acids, which are just 3 carbon compounds. We break the glucose essentially in half and produce 2 3-carbon units, plus 2 NADH. The NAD must be present to capture the electrons, the high-energy electrons, at a particular step, and 2 ATPs. You need to have some ADP present in order to phosphorylate and make ATP. So, usually when we're operating in the isolation, glycolysis is happening at a very high rate in some muscle cells that are specialized for this process. This is what they do. This is how they make energy for contraction, just anaerobic glycolysis. So it's going along at a very high rate, and relatively quickly, NAD, the supply of NAD in the cell, is all converted to NADH. Right? Keep capturing electrons, and we don't have any more NAD. And so now glycolysis can't continue, were that the case. But it's not the case, because these cells that are specialized for anaerobic glycolysis will undergo fermentation. That's a process by which the electrons and hydrogens on the carrier are transferred to pyruvic acid or pyruvate, forming lactate and restoring NAD. We're going to clear those electrons off the NAD so we can bring it back up here and do more glycolysis, run more glucose molecules through that process. So fermentation, in order to continue a high rate of glycolysis, is the reason for lactic acid production and that familiar fatigue process. Here's a little chemical diagram of this happening. Pyruvate can accept electrons and hydrogens from NADH, restoring NAD and forming lactic acid or lactate. Lactic acid is the compound with the hydrogen still attached onto the oxygen, and so here's lactic acid, and then acids are those compounds in our definition which can release hydrogen ions into solution. And so here, once that hydrogen ion dissociates from lactic acid, we have lactate. So that's why you keep hearing me say sometimes lactic acid, sometimes lactate. These are interconvertible forms in water at the pH of our bodies, and so that's why. And here's a little diagram showing the energy investment portion of glycolysis, right? We start with a glucose molecule. We have to phosphorylate it, and then again, using up 2 ATPs. But then as we oxidize it through a number of steps, forming 2 pyruvates, we get out 4 ATPs, subtracting, you wind up with 2. So the net ATP production is 2 per glucose, and ultimately we get 2 NADHs per glucose. One thing we should talk about, usually muscle cells are using glucose stored in the form of glycogen. So in the muscle cell will be some glycogen, which is just a big polymer, big collection of glucose molecules all concatenated together into a big tree-like structure. And in order to make glycogen, glucose molecules first have to be phosphorylated to form glucose 6-phosphate, uses up an ATP. And then we need another ATP in order to store that glucose 6-phosphate in the form of glycogen. So another ATP for every glucose that's stored as glycogen. Then when we bring the glucose back out again from glycogen, convert it back to glucose 6-phosphate in order to undergo glycolysis, here's the second ATP from the quote energy investment portion of glycolysis before we can proceed. So we have to burn or utilize an extra ATP per glucose if we want to store it first and then use it for glycolysis, right? That's what this arrow's downward arrow represents glycolysis. Glucose, pyruvate. Another way to think about that is if I could say, it might be even more clear if I say it this way. Glucose is first phosphorylated as we bring it into the cell and prepare to use it for glycolysis, and then we add one more ATP, and then we can run it through glycolysis. So 2 ATPs of the investment phase of glycolysis, and then 4 ATPs are released through the energy generation phase. So this is just a depiction of what we were already talking about in the previous slide. But then if we store the glucose first as glycogen, we have to invest another ATP for every glucose. So when we're figuring out our total tally at the end of glucose or ATPs produced by glucose, we need to take that into account. We have already mentioned the electron carrier molecules, NAD and FAD, so I just wanted to remind you, it's an obligatory step during glycolysis, and also, as we'll see in a bit, in the mitochondria, we must capture the high energy electrons released and form NADH. And we must capture some electrons at another step in the mitochondria and form FADH too. So that's what's going to be our deal if we want to make even more ATPs than we can make by glycolysis, and we're doing it at a more submaximal exercise rate, then we can produce ATPs by aerobic respiration in the mitochondria. But as we'll see in just a second, we're going to get a lot more ATPs for every glucose, and we are not going to need to produce any lactic acid. So that's the beauty of this. Aerobic respiration occurs in the mitochondria. We do initially run our glucose through glycolysis. We're just going to now add, in addition to that, 2 more series of reactions: Kreb cycle, or the citric acid cycle, and the electron transport system or chain. So in Kreb cycle, we take the pyruvate, modify it, and then oxidize it right to CO2 and water, and we'll produce an ATP for every time around this cyclical set of chemical reactions, but the main thing is we'll produce high energy electrons in the form of NADH and FADH. Then we'll take all the high energy electrons, both from glycolysis, right? Remember we had the 2 NADHs forming glycolysis. We're going to shuttle those into the mitochondria, and we're going to produce NAD and FADH in Krebs cycle, shuttle all those electrons to something called the electron transport chain. And we're going to use the energy from those high energy electrons to make ATP. So this is the skeleton of what we're going to do to make a lot of ATPs per glucose by aerobic respiration. Hans Krebs was the biochemist who discovered the series of chemical reactions that we now call Krebs cycle, or the citric acid cycle. This way of taking the pyruvates from glycolysis and oxidizing them completely to CO2 and water to get more high energy electrons from them in order to be able to produce a lot of ATP by oxidative phosphorylation, it's called. So in part 4, we'll look at very closely and in detail at the processes happening in the mitochondria, both the Krebs cycle and oxidative phosphorylation, this method of using the electron transport system to get the energy from the high energy electrons. And we'll see how we make ATP that way. We'll also look at the efficiency of ATP production by aerobic respiration.